I'm very happy to introduce Professor R. Sowdhamini. Is a senior professor at the National Center for Biological Sciences, NCBS, Bangalore. She holds a prominent position in the field of computational biology and is known for her pioneering work in structural bioinformatics and systems biology. Her research primarily focuses on the structural bioinformatics, protein domain, superfamilies, and plant genomics by transcriptomics. Professor Sowdhamini has contributed significantly to the development of computational tools for predicting protein structure and function, as well as for understanding the role of macromolecular interaction in cellular functions. Her work integrates computational models with experimental data to provide insights into various biological phenomena, especially in the context of infectious diseases and cancer. She has published extensively in international journals and has received several accolades from her contributions to science, including the fellowship of the Indian National Science Academy, INSA, Indian National Science Academy. She has received several prestigious recognitions, including the Human Frontier Science Program Award, International Senior Fellowship from the Wellcome Trust, and currently a J.C. Bose Fellow. Professor Sowdhamini has also been actively involved in promoting science education and collaborating research at the national and international levels. So we have two JC fellows addressing us. That is a very nice thing to have. Thank you very much, madam.
So our lab has, as rightly been introduced, our lab is dedicated to structural bioinformatics, and we got interested in plants early on, and our interest in how plants respond to stress, being a very broad thing, has been there since 2004, and some of you have been following our lab interest. And what we have been asking is rather like how Usha had given a very detailed and nice beautiful presentation, namely looking at transcription factors and how some of them are vitally involved in my case, stress response. Whereas today what I thought I will talk about is a very specific sort of a stress, a biotic stress, which is the fungal attack by a particular plant. So this is like one type of stress, one type of plant, and we are not even asking what are the transcription factors involved, but instead using bioinformatics tools to look into the huge amount of transcriptome data that can be obtained at different stages of this particular fungal attack. And a plant that has been chosen for this description is pepper. So the title is somewhat general, saying how do plants defend against plant attack, as it should be fungal attack, sorry. And the specific plant under consideration is pepper, and the fungus that we are talking about is Phytophthora capsicum, and this was mentioned in today morning as well. So, this being the case, let us discuss about the plant. It is not a model organism, but it's a very commercially important one. It's a spice crop. It is mentioned, it's used a lot and grown a lot in Kerala, and it requires certain climatic conditions and also long trees so that it can just climb around it. So the conditions are suitable in places like Kerala, although it can be grown in other places as well, in Tamil Nadu as well. And in fact, in a part of Tamil Nadu, it has a different variety, a stronger variety. But in the context of the fungal attack, what are we interested in? It is a disease called quick wilt disease that is induced by the attack of this particular fungus, and it can give rise to nearly 30% of the mortality in this plant. And there is also a specific variety called the Panur variety, which is known to be susceptible to this particular gene, this particular fungus. And what we are going to discuss today is how is the plant getting ready in terms of its defense mechanisms when the plant is subject to this particular fungus, right? And for this, we will take the example of the Panur variety, because that is more susceptible than others. And this particular work has been done in collaboration with an institute in Kerala, Rajiv Gandhi Research Institute in Trivandrum, and the The lab that I am talking about is Dr. Manjula's lab, and they already had grown the plant at different times of infection, namely after 3 hours, after 6 hours, and after 12 hours. And the plant would start to succumb to this, as we can see, as early as 6 hours after infection by the fungus. And you can see the fungal growth that is happening, the hyphae can be evident at the time of 12 hours post-infection. So just like how Usha had mentioned, like different time points, you extract, purify the RNA and then do an RNA sequencing of it and then compare them. This is called as a cross-transcriptome analysis. So we do this at different time points, and we note that 3 hours per infection because there is so little difference between the 0th and the 6th one. So we took 3 time points, the 0 hours before infection, namely the wild type without any fungal attack, and then the one at 6 hours and one at 12 hours. And you can see that it is quite evident at 6 hours there is a microscopic growth of the fungi that one can observe. And then the infection just proliferates and you can start to see neurotropic factors coming in by a day. And it was not done at the end of 24 hours because there is a whole lot of side effects that were going on in the plant, lot of cell death going on. So we did not want to have interference from the cell death factors. So that's why we chose the 6 hours and 12 hours post-infection. As mentioned earlier, the RNA would be purified from them and then given for transcript sequencing. And we were left with a lot of transcript sequencing data, and this is usual. You can see millions of reads, nearly 81 million reads for the control, which is nothing but the what we might call as the 0 hours post-infection, which is nothing but a wild type in this case. And then after 6 hours after infection and 12 hours after infection. Every time you're dealing with like millions of reads, 57 million reads and so on. And fortunately for us, you may say, but halfway through our analysis, the genome of this particular pepper plant became available. In fact, not only the genome, but the transcriptome assembly was available. So we used that as a reference and then used these 3 reads. And to begin with, we just combine all the reads as if it does not matter, because after all, the transcripts are all from the same plant, very same species. So for the sake of transcriptome assembly, we just combine all these reads. So you get more and more reads to begin with, and then use these referenced assembly as a guide to come up with the assembly of this particular plant. And then we can backtrace. After you get a robust assembly, we can backtrace and ask different questions. And one question we would ask is, how many transcripts are there in the 6 hour post infection, which is not there in the wild type? And same way, how many transcripts have been newly expressed or downregulated after 12 hours of the infection. So we are going to keep looking at the upregulated transcripts and the downregulated transcripts at both time points. And this is always done with respect to the wild type. And just to give you statistics, the combined assembly has very good N50 values. If it is there in the order of thousands, that means it's a good sign. It's nothing but if you were to put all the contigs that you have just built up in the descending order of its length, at 50% of the length, how many nucleotides are we able to see? So if you get lot of little fragments when you bring in the transcriptome assembly, that means your N50 value will be less. Because even if you put them in the descending order, at 50% cutoff, you are able to see little fragment, but higher the value, therefore it is a good assembly. And anything in the order of thousands is considered quite good, and we have about 2,500 base pairs at 50% cutoff. So then, like I said, getting the assembly is just the start point, and then we would always do what is called as a comparison. In this case, wild type versus 6 hour post-infection, and a wild type versus 12 hour post-infection. And this is called as the differential expression, or what we might start to call as DEGs. DEGs are not the essential genes, but in this case we mean the differentially expressed genes. Either they may be upregulated or downregulated with respect to the wild type. And to begin with, of course, we have to understand if the assembly is good enough, what sort of domains are seen in the assembly and so on. And for this, function annotation was performed against the entire plant database, Viridi plantae. And we noticed that the plants that are closest are the ones like cinnamon and lotus. And we know, how do we know this? Because every transcript that we have been able to identify in the pepper plant, we look at the nearest best homologue when you do a sequence search. And if many times we get these the homologues from these species, that means we conclude that these are evolutionarily closely related. That does not mean this is the closest from the Darwinian angle, it's just the closest plant for whose sequence information we have available in the Viridi plantae database. And then we noticed that when we start to look for the function annotation, kinase containing domains and then likewise ubiquitin degradation domains as well as the PPR domains, pentopeptide repeat domains were highly expressed in this assembly. And each of these are quite important because it may be a little biased, because after all 1/3 of it is wild type and the 2/3 are during the course of infection, fungal infection. And we can relate because kinases are required for carrying on the signal transduction. For telling the phosphorylation is viewed like a switch that tells other genes to be expressed or not. And then we have ubiquitin related proteins because that completely regulates the turnover of the proteins because it can give rise to degradation. Whereas the PPR proteins are potentially the post-transcriptional regulatory proteins, and they are also highly expressed. And plus we also noticed other domains that are known to be nicely binding to the RNA themselves, the RNA binding proteins, RRMs, and as well as those domains that are known to bind to other proteins like LRR, the repeat containing domains. But on the whole, we could see that when we search against another database, which is that of PFAM, which is called as PFAM, but these are nothing but proteins whose homologous sequences are available, and we know at the domain level what are all the homologs. So these also suggest that there are quite a lot of these PPR containing domains that are involved in post-transcriptional regulation, as well as the LRR domains that are there to recognize foreign molecules, foreign RNA or foreign lipopolysaccharides and pathogen especially. So these sort of genes are getting highly expressed, and this means that the plant is responding to the presence of this fungal pathogen and getting its own immunity primed and kept ready. So if we were to take the wild type transcript and compare with the same plant, but after a few hours of the infection, we can see that quite a lot of genes are getting upregulated during the infection, and that is shown on the right hand side. It is called as a volcano plot because you're comparing the transcript, the number of reads that get aligned to each of the transcripts that have been recognized by the assembly. And then we are comparing it with how it is in the wild type versus 6-hour post infection and 12-hour post infection. And whatever is on the right hand side are highly upregulated genes, and whatever is on the left hand side are getting down regulated upon infection. Okay, so this is very important start point because that's where we can begin to start ask specific questions. For example, if we focused on the differentially regulated genes, the upregulated genes are those which you can see in high values. That higher means the transcript, the millions of reads that get aligned to the transcript, if it is more, then the shade is going to be dark blue. And if it is downregulated, that means fewer RNA pieces are getting aligned with the transcript of interest. You will see that very light color is there. And what you can see straight away is in compared to the wild type, where there is no infection going on. I should say wild type, but it is really a control because you're not changing the gene here, just putting it through infection. So control has a fewer number of RNA pieces that get aligned to the transcripts. But in relation to that, upon infection, there is a huge abundance of RNA reads that get aligned to the transcript. And what sort of genes are they? Many of the upregulated genes happen to be the methyltransferases. You can see methyltransferase type 1, type 2. So these are a different type of methyltransferases. And you can see that they are getting upregulated during the course of infection. And further bioinformatics studies also suggest that on the other hand, whatever transcripts were highly abundant in the control are poorly expressed, poorly transcribed during the course of infection. And this you can see because the dull color means lower number of RNA pieces are getting aligned. And what sort of genes are these? These are mostly transcription factors, photosystem related genes and cell wall process associated genes. So basically any synthesis machinery that is to do with building strength or building more genes seem to be silenced. And this is almost like, when the plant is under stress, it is not going to expand this energy by means of producing more and more genes that are not required. As an analogy, we can think like when the demonetization was announced, people were so busy getting all their cash and putting in the bank. Hardly anyone went and withdrew more cash, right? So it was not even possible. I'm saying the same way, if there is stress, a biotic stress such as fungal attack, the plant is busy getting all these regulatory mechanisms in place, but it will not be spending its energy producing more genes than is required, or to get into all these photosystem related genes and so on. So it's almost in a freeze mode, and it would do just enough to save itself. So that is the exact question that we want to ask. And we can also ask this in a different way and say there is a way of enriching what sort of functions are there, whether it depends on the biological process or the cellular compartmentalization or the biochemical activity. You can classify genes, or in this case transcripts, into very different types. And function of a gene in the bioinformatics term is a very hierarchical term. So that's why these three broad categories are there. Whereas most people think of biochemical activity as a function, you can also think of it in which pathway is it involved and which part of the cell it is involved and so on. So time and again we notice that the metabolic processes and catalytic activities and those sort of transcripts are highly upregulated during the fungal attack. On the other hand, any production such as cellular processes and DNA binding properties are getting downregulated. Okay. So this being the broad sort of realization, like new processes are all stunted, and then the basic defense mechanisms are going on. We just quickly asked questions as to the well-known cherry pick pathways such as the salicylic acid, jasminic acid and the ethylene mediated signaling pathways. And for this, there are many genes involved. In the interest of time, I would not be going through all of them, but the salicylic acid and the jasminic acid we found were key signaling pathways that get upregulated during the pathogen attack. The salicylic acid would get upregulated 6 hours post-infection more. And why? How do we know this? Because we have transcript data at different time points of fungal attack, right? So the salicylic acid ones are the early responders. And then the jasminic acid signaling pathway members, as we can see from the transcript data, are late responders. Late in the sense at 12 hours. Instead of 6 hours post-infection, you start to see those pathway genes that are highly expressed 12 hours post-infection. On the other hand, the ethylene signaling pathway genes are a little bit of a mix because they go through a dynamic process where they themselves are regulating both other pathways, like namely the salicylic acid and the jasmonate one. So how do we know? So we use a protocol that we have developed in our lab by which if you are looking for a gene of interest in a big assembly or when you are doing cross-transcriptome analysis, what we do is to start off from a similar gene from a closely related organism, in this case plant. And so that means if you are interested in a particular gene involved in the salicylic acid pathway, so you pick a similar gene in a closely related other plant and then use that as a query, and then where possible you accumulate homologs, meaning not only in the closely related plant but the same protein but in multiple other plants. And what we have to make sure that these are all authentic members, trusted homologs, and for that we do a clustering of phylogeny by which we get to see members that are probably belonging to that broad family, but they are getting to cluster themselves away. So you can remove some outliers that you don't want to put in your query, and then you basically create a multiple sequence alignment and use that as a jump start into then do a sequence search into our transcriptome assembly or in the particular set, which may be 6 hours post-infection or 12 hours post-infection. And then the hope is that we will start to pick up transcripts that belong to the plant of our interest. And it does happen, and it is a good way of recognizing the close homologs, and we propagate, do a multiple sequence alignment, do a phylogeny, and make sure that the newly added members from the plant of interest are indeed co-clustering and they are not outgrouping themselves, which suggests that there could be a very sensitive approach of sequencers. Therefore, we are picking up many that don't really belong to that family. They are not necessarily false positives either. Okay, so once we have that, we can bring in and do a meta-analysis, bioinformatic analysis, by which you then ask the question in which tissue or which condition are these getting upregulated, and does it agree with the known biology that has already been evident for those systems. So if it is not there, it could be a new learning that we can perform. Okay, so we asked this for different signaling pathway genes. Like I said, the salicylic acid, where a number of proteins are involved, and unfortunately I won't be able to talk about them. Each one of them has a story behind and how they specifically help with the fungal defense. And we can see from the TPM, which are nothing but the number of millions of reads, RNA reads that are getting aligned to the transcripts, that, for example, the genes in the salicylic acid pathway are highly abundant 6 hours post-infection, as opposed to the control, or even opposed to 12 hours post-infection, suggesting that the genes that are in the salicylic acid signaling pathway are triggered first in response to the fungal attack. On the other hand, if we closely follow the genes that are involved in the jasminic acid signaling pathway, we see that the expressions are not exactly having the same trend. You see higher values 12 hours post-infection, suggesting these are late responders. And we can do this for the members in the ethylene signaling pathway where there is a mixed response. So that suggests that the ethylene signaling pathway, for example, it's itself modulating the defense response of the plant to the fungal infection. And these have been validated by means of RT-PCR, so which has been done at the collaborators' end, suggesting that whatever we see from the transcript and those trends are indeed validated using these sort of specific experiments, where the plant is subject to those very same conditions, and then we look at a particular gene of interest and find out whether their expression is also high. And you can see that the overall similar trend is there. And on the whole, it also suggests that if we have a manner by which you can purify RNA and you can follow a plant through a particular fungal attack, there is a possibility to recognize the key players, whether they are transcription factors or well-known pathogen defending genes. You can begin to ask very specific spatio-temporal questions. In this case, there is no tissue level differentiation. We just take the plant leads, but for another plant system, the question could be much more fine-tuned, and in this case, it's mainly temporal. And then we get to understand, for instance, depending on the pathway, different genes get upregulated at different points of infection. And these are the people who have helped me through this project. This is Dr. Manjula and her student in GCB, and the particular pepper fungal attack work was carried out by two of my colleagues, Dr. Shafi and Dr. Nagaratnam, and the work was funded by the DPT in terms of a specific project. With this, I'll stop and take any questions you might have. Thank you.
Thank you, Professor. Quick round of questions. Yeah, you go ahead.
My question is, the study was conducted in Paneer 1, which is actually a susceptible variety. And we saw that methyltransferase genes were upregulated and many others related to synthesis were downregulated. Can we expect the exact opposite trend in resistant varieties, or can we expect some variations in the trend?
Yes, yeah, that is a very good question. And in fact, for us, we always say the project never ends, because that is the next thing we would like to do, given a lot of funding, because you can see how many raw reads are required for us to get a good transcriptome assembly. So there is indeed a resistant variety. And the farmers have already been dealing with this sort of a morbidity issue by means of hybridization. and it seems to work very well. So that, and people have noticed certain genes are down-regulated and up-regulated that follows this trend, but we have not taken the naturally tolerant variety so that we can say with a higher assurance that it concurs. But for that, it will be a next part of the study, I suppose.
Thank you, ma'am.
Yeah. Nanopore is more suited for genome level assembly.
Yeah.
Again, yeah, we can do that, but the problem is, you get long length DNA, but it's also error prone. So you
Now the error rate is coming down
it's
quite
coming down.
drastically.
Yeah.
If you
So
do
again, if we have the funding, why not?
if you
Yeah.
do 1D square, then it will
Because
be a lot.
because it will really help us to know what are the transcription
Right.
factors that are upstream, and that kind of goes with our original
It will
interest.
also be easy to assemble, and
Yeah.
you don't actually need to assemble them in that case. You have a long read, so you'll have the isoforms also easily.
Well, theoretically, yes.
identified.
Yeah.
Practically also, I guess.
Yeah. That.
Yeah. That is when there's the method rate. So mostly you combine.
Yeah, we get data in our hands. It has happened as low depth. Yeah, that's the problem. With these long reads, the depth is low. But if we are looking at non-model organism, that may be good enough.
First enough, thank you, ma'am. Ma'am, you mentioned that the upregulated genes are like the defense genes, but sometimes it may act as a negative regulators for that. What's about that, ma'am?
Negative regulators
For that organisms, because I'm worked on Isotonium solani in rice, it creates some effectors and the transcription factors. It sweet genes are act as a negative regulators for that. So
Yeah. I mean, definitely, it's possible that some of the genes are there just to regulate the upregulation of others. So, there are many layers of regulation, and like Usha also said, it's like feedback, feed forward regulation. It's very complex. So, unless and until we understand the biology of the genes, we cannot infer what it means. So, we have to work hand in hand with the biologists, or at least understand the biology of these systems. Because without that, these are just numbers, so it wouldn't be fun.
another question, I missed that. Why you not take the 24 hours of after post inoculation? Why you take only 6?
There the problem is, first of all, practically there is a sample handling problem because a lot of it is just already dying. And the second is that a lot of the cell death regulatory cell death genes are getting involved. For example, all these BCL and those sort of genes would start to come in the way. However, we really want to see how the plant is trying its best to defend, because we don't want to really see when it is succumbing to the pathogenic attack, right? Because we want to know how it is trying to not die.
Because after 24 hours, it may overcome from this pathogen.
Possible, but this is a susceptible variety, so
Yeah.
unfortunately,
You can discuss with madam during the
Okay.
coffee break.
Yes.
That will be more.
Okay, sir. Thank you.
Yeah. One more question we can take from the students. On one side, you elaborated about the defense mechanism in the plant, but from the fungal side also, if we really look at how they,
Yeah, correct.
that may be of.
Yeah, actually, the
Yes.
perspective is very different. Fungal perspective, Amai is looking at it. One of my
I
colleagues.
was just going to say that in that very same data set, you can actually query to see how many transcripts are actually mapping to the fungal.
Fungal
You
genome.
don't have to do a different. You have to,
with the current
in
data.
the same data set, you should be able to do it.
With the current data set itself, we should be able to do. And in fact, one of my colleagues, Amay Redkar, is more interested in the fungal perspective. And for instance, what are the hyphae doing? How does the plant give itself to the fungal attack? And the interface between the two is also interesting. The plant-fungal interaction.
Yes, advances in fungal biology is also now equally understood, and it's easy for us to compare these and see where exactly
And we heard in the
they
morning
meet.
also. Yeah.
Okay, thank you very much, and